we're going to talk about muscles.
Bill Nye the Science Guy. Bill Nye the Science Guy. Bill Nye the Science Guy.
Science rules.
Bill Nye the Science Guy.
Inertia is a property of matter.
Bill Nye the Science Guy.
T minus 7 seconds.
Bill Nye the Science Guy.
Hi!
We're going to talk to you today
About...
the 3 different types of muscles.
Let's begin. First, we're going to talk about the 3 different types of muscles.
Well, we have 3 different types of muscles that are found in the body. The first is cardiac muscle. This is only found in the heart. The second is skeletal muscles. This will be found attached to your skeleton or bones. And the third type is smooth muscle, which will be the rest of your muscles inside that cover your visceral organs, excluding the heart.
Tell me about the cardiac muscles, Tina.
Sure, Glenn. You see, cardiac muscle is exclusively found in the heart. And those cells are relatively small, and they have a single centered nucleus. In addition, cardiac muscle cells are branched, and they make contact with lots of other cells. So, this allows for rapid movement of molecules from cell to cell, and also allows for action potentials to occur quickly. In addition, cardiac muscle contractions last 10 times longer than skeletal muscle contractions.
Smooth muscle, the second type of muscle, is found primarily within the hollow walls of visceral organs. It is involved in processes such as peristalsis, which involve involuntary movements. It is spindle-shaped and, generally speaking, long, and this unique structure allows it to carry out these processes.
So, the third type of muscle that we're going to be looking at is skeletal muscles. Skeletal muscles help you to move your body, and they have multiple nuclei per cell and are striated. They are pretty unique and help you to move your bones.
Bang!
Let's learn more.
Have you ever wondered how the muscular system interacts with the skeletal system? I know I have. Let's look into it.
Now that Allie and I have completed such a wonderful task, I think we'd like a good high five. There we go. Now, in this high five, what exactly just happened? Well, let me tell you, a lot is going on. As we talked about before, you have the brain interacting with the muscles. After this happens, the muscles themselves have to interact with its skeletal system. Whew. Once this happens, the muscles will pull on each other, and when they're pulling, they're also connected to the bones from tendons. At these tendons, the bones are connected at joints. When the tendon is pulled, the joint itself will also move, allowing for what we see as contraction on the outside. When this happens, it allows for a lot of activities, such as dual-wielding ping pong paddles. Let's move on.
Hello and welcome to Cooking with Christina. Today we're going to saute up some sarcomeres and grill up some cells. Let's get started. For today's craft, you will need a few simple ingredients. Straws, saran wrap or stretch tight wrap, a pair of scissors, and a cardboard tube. That could be a paper towel tube or a toilet paper tube. So today we are going to be making a cell. So, okay, get started with your tube. You got your tube. You will take saran wrap and cover the outside of this tube. This covering will represent the epimyocin. Next, take your straws and cut them in half, like so. Excellent. Okay. After you cut up your straws, you can put them into bundles. These bundles will represent the muscles, the bundle of muscle cells. Then you can put them inside your tube. Each of these muscle bundles are surrounded by saran wrap, which is the endomyosin. If you look inside, you can see how these are represented. This is how it looks like in a muscle cell, except they're filled all the way. Let's look at our craft one more time. We have our muscle. Then we have our muscle fascicle. From the muscle fascicle, if we look even deeper, we have the skeletal muscle fiber cell. Within each of these muscle cells, we have transverse tubules, which are filled with extracellular fluid, and they're like tunnels throughout the mountain. Also, each skeletal cell is made up of functional units called sarcomeres. These are thick and thin filaments. Myofibrils are bundles of myofilaments, which are made up of actin and myosin, which we'll talk about next.
The muscular system interacts with many other systems. For example, when a muscle contracts, it requires the interaction of the nervous system with the muscular system to do so. First, a signal will be sent from the brain through the nervous system, through a nephron, into a neuromuscular junction, which is right at the surface of a muscle cell. It is at this point where muscle contraction actually occurs. We're going to talk to you about that just a little bit more. Woo! Now, within a single muscle cell, we are within the sarcomere. I would be the myosin, and this is the actin. As you can see from the arms here, the arms represent the tropomyosin, which is covered by the troponin. So, at the neuromuscular junction, once acetylcholine, a neurotransmitter, is released, it will indirectly cause also the release of calcium, which will in turn expose the active site for the myosin head to attach. Now that my myosin head is ready to attach, ATP will hydrolyze, leading to ADP plus PI, hooking on to the actin. From here, the troponin will pull the muscle, causing contraction.
Well, friends, this concludes this week's episode about the muscular system. Join us next week as we explore other body systems.
Biceps, brachii, triceps, brachii, origin and insertion, where it starts and ends. Orbicularis, deltoid antagonist is a muscle that contracts when one relaxes. You can hear platysma, pectoralis major, abdominal, tendon, and next muscle to bone. Gluteus, maximus, gastrocnemius, fascia, between organs and muscles. We know all our muscles. They are always pulling and they're never pushing. We know all our muscles. We can label them all and we can ace our finals.